Hello everyone and welcome to Instant Biology by Dr. Neelab. The topic that I am going to start today is complement activation pathways. So if you have been following my lectures, then you would know that I have already made a lecture in English about basic complement system, right? So in that video what I explained was that what is complement system and basically what are the functions of complement system, and in that video I made a connecting link that I would be talking more about the different pathways through which the complement system can be activated. Now, as I've already said, that complement activation or complement system activation is the last resort. That means it is one of the final things that a body can do in order to safeguard itself, right? So therefore there should be very stringent measures, there should be very regulated measures through which a complement system can be activated. So in this particular lecture I would be talking more about that what are the pathways through which this complement system can get activated. So talking about the first pathway. What is the first pathway? It is about classical, or it is the classical pathway. So the first pathway, the name of the first pathway is classical pathway. Why is such peculiar name given to this pathway? Why are we calling it as classical pathway? The reason behind this is that it was the first pathway to be discovered. So this is the first point that you can see on your screens, that it was the first complement pathway that was discovered. That is why it is called as classical pathway. The next important thing is that how does this classical pathway start? Now understand this, that you all would be knowing about antigen antibody interactions. I have made multiple videos on antigen antibody interactions, so you would be having a clear-cut idea on the same. What happens is when antigen and antibody interact with each other, what happens is complexes are formed, right? When the size of the antigen is large, or when the number of antibodies that are interacting with the antigen are large, then what happens is the antigen antibody, they interact and form complexes. These are large complexes, right? So these large complexes, these antigen antibody complexes, what are they doing? They lead to the formation of, lead to the activation of classical pathway. Okay, so the first thing over here is clear, that classical pathway gets activated because of the antigen antibody interaction or the formation of antigen antibody complexes. So this was the second point on your slide. Talking about the third point, if someone asks you that what or how, or let us reframe the question, that what does or which wing or which part of your immunity does the classical pathway serve? So you might be knowing that there are two parts of your immunity, right? Adaptive immunity and innate immunity, right? Innate immunity and adaptive immunity. So innate immunity is the immunity that you get by birth. Adaptive immunity is the immunity that you adapt, that you get in the process of your life, right? So if I'm talking about this classical pathway, this classical pathway is actually serving the adaptive immune response, and if you are going in much detail in the adaptive, it serves the humoral immune response. Now, for those of you who do not know what humoral immune response is, in the adaptive wing or in the adaptive class, adaptive immune responses, there can be two types of responses. One can be cell mediated and one can be humoral response. Humoral immune response is basically mediated by antibodies, whereas the cell mediated responses are basically regulated by the T cells, okay, T helper and T cytotoxic cells. However, I am not going into the details of the humoral and the innate systems. I have already talked about that in one of the previous lectures. So When I'm talking about the effector pathway, that means which pathway is this classical system, or which part of the immunity this classical system is serving, classical pathway is serving, then you can say that it is serving actually the humoral adaptive immune response. Humoral, that means it is being mediated by the antibodies, right? Now over here in the second point itself, it is written the antigen antibody complexes. That in itself means that antibodies are involved over here. If the antibody is involved here over here, then definitely it will be somehow related with the humoral adaptive immune response. I believe you would have a clear understanding about this now. Coming to the last point of this slide, what are some of the other activators of this classical pathway? So the other activators might include viruses, necrotic cells. Necrotic viruses you already might be knowing. Necrotic cells means the cells that are dying, the cells that have that are in the process of dying or they have died. They might also activate the classical pathway. Subcellular membranes. What does subcellular membrane means? So the membrane that is present inside the cells. Now you would be knowing that there are multiple organelles that are present inside the cells. For example, mitochondria, chloroplast, these are the organelles that are present inside the cells. Now if inside the cells these membranes, the membranes of these organelles, for example mitochondrial membrane or chloroplast membrane, it undergoes some abnormality, then this might also serve to to activate the classical pathway. Next is aggregated immunoglobulins. What does that mean? Immunoglobulins is a more technical term for antibodies. So what happens is when multiple antibodies are aggregated together, when multiple antibodies have joined together, it will also lead to the activation of classical pathways. Now let me just introduce over here one more important thing, that whenever the immunoglobulins are aggregated, it is mandatory to understand this, that there are not many immunoglobulins have the capacity to aggregate. You might be knowing about IgM, immunoglobulin M, right? So this immunoglobulin M has a valency of 5, pentavalent, right? Now this means that it can it can interact with multiple things at one time. So that is why IgM has the highest capacity basically to interact at multiple places. So therefore it has the highest ability to activate the classical pathway. The next thing is amyloid, beta amyloid found in the Alzheimer disease plaques. Now you would be knowing that Alzheimer is a disease in which a person loses his or her immunity. And why does the memory. So why does the individual lose his or her memory? Because of the deposition of beta amyloid proteins inside the brain, right? So this deposition actually interferes with the capacity of the brain to function, and therefore a person loses the memory. Now this beta amyloid protein, apart from disturbing your memory, it also can activate the complement system. So these are some of the important points that are related to classical pathways. Okay, one more important thing I would like to add up over here. I should have said that before starting classical pathways, but just now I remembered it. We should know what does a complement system do. In general, the function of complement system is to aid in opsonization, or to aid or to facilitate in the phagocytosis of a pathogen. Okay, so whenever we want to phagocytose, whenever we want to engulf a given pathogen, then the complement system comes into activity. It does not engulf by itself, however it is aiding the other cells, the other phagocytotic cells, to engulf the said pathogen. Understood? I believe you would have understood this clearly. So this was a little bit introduction about the classical pathway. Let us move on to the next slide and understand how does this classical pathway function. Now, as I've already said that this all starts with the formation of antigen antibody complex. Now on your screen you would be able to see that this antigen antibody complex is present, and on this antigen antibody complex a complement protein comes and binds. The name of the complement protein is C1. Now I have already told you that complement systems are named like C1, C2, C3. So this is one way of naming. Some other names can be on the alphabets, for example factor B, factor D, like this. So let us come back to the discussion. When the antigen antibody is formed, when the antigen antibody complex is formed, what happens is this C1 protein comes and binds to the antibody portion of this antigen antibody complex, right? So it is just binding the antibody portion. Okay, now one more thing, this C1 is actually made up of this complement protein 1 or C1 is actually made up of 3 subunits, or you can call 3 portions or 3 parts. What are they called? C1Q, C1R and C1S. So this is just alphabetic in nature, Q, R and S. So this can be a trick, you can easily remember this, Q, R and S. C1Q, C1R and C1S, they are parts of the C1 protein. And if I come to C1Q, if I talk about C1Q, it is made up of 6 identical subunits. If I talk about C1R, it is made up of 2 molecules, and same with C1S, it is also made up of 2 molecules. Now this C1, as I've already said, it is binding to the antibody portion of the antigen antibody complex. Okay, so by which of these C1Q, C1R or C1S does the C1 protein bind to the antibody? Okay, so the question arises, with the aid of which one, C1Q or C1R or C1S, which one amongst these aids the C1 protein to bind with the antibody? So the answer is C1Q actually aids in binding. You might be knowing that, let me just draw over here, you might be knowing that antibody has this kind of a structure. Okay, and this region is called as the FC region. Okay, and this FC region is actually involved in interaction with the complement. That means C1Q would be binding over here. So let us say C1Q is binding over here, and along with C1Q comes C1R and C1S, that is making the entire protein. Let me just draw this, C1R, C1S. So this complete thing is called as C1 protein, and together dividing it into 3 parts, C1Q, C1R and C1S. So now you know what is the prime function of C1Q. C1Q is involved in the binding of the C1 protein with the FC region of the antibody. What about C1S? Now let me talk about C1R and C1S. So what is the function of C1R? C1R is actually proceeding, or it is actually involved in the cascade or signaling cascade. So C1R is actually a serine protease, and it is involved in transferring the signal. It is involved in activation cascade. That means it will activate C1S. So what does C1R do? It will activate C1S. And what will C1S do? C1S will cleave C4 and C2. Okay, so what will this C1S do? C1S will cleave C4 and C2. Now you would be knowing what does whenever the cleavage of a particular complement protein happens, then what does it form? It will form 2 portions, 2 parts, A part and B part. So over here this C4 will be broken into C4A and C4B. I have already told you that the A part will move away. In the previous class I have said that A for away. So this C4A will move away, whereas this C4B will stay at the point where it was activated. Okay, so I have written also this C4 will move away, whereas C4B it will stay. Similarly, this C1S will also cause the breakage of C2. The C2 will again break into C2A and C2B. But I said in the previous class only that C2 serves as an exception, because when C2 breaks, C2A and C2B are formed all right, but C2A will not move away like the other complement systems. C2A will stay and C2B would go away. So this is an exception. In all other cases you would say that the A part will move away, but in only C2 complement you would say that C2A will stay, whereas C2B will move away. So the functions have interchanged. What does the A portion do? When it goes away, what does the A portion do? Actually, the A portions are involved in the inflammatory responses. Okay, so the A portions are involved in the inflammatory responses, and B portions, they particularly stay. For example, C4B will stay over here, and in case of C2, C2A will stay over here. So they will regulate, or they would do their thing of activation of the complement system. We would talk more about that. So if we return to our discussion over here, C4B and C2A, they together form the C3 convertase. Now I said that C4B will stay over here and C2A will also stay over here. So the combination of C4B and C2A together is called as C3 convertase. This is important. Why is it called C3 convertase? Because it would be converting C3 into its two parts, that is C3A and C3B in future. We would learn about that in the coming slides. But for now you need to understand that C4B, C2A is responsible, or it functions as C3 convertase. In this, it is acting on C3 in order to break it into C3A and C3B. So over here, this is the portion where we end the classical pathway, the formation of C4B, C2A. So it can be written like this also, C4B, C2A, and it can also be written like this, C4B, 2A. Right? That means you do not need to write every time C, C, C again. So you can just write C once, C4B, 2A. Now this is activated. Whenever something is activated, you can just put this bar above the name of the complement. It will indicate that it is activated. When it is activated, it will act on C3. When it acts on C3, C3 gets broken into two parts, C3A and C3B. Therefore, it is called as C3 convertase. Now, over here we end up the classical pathway. I have a particular diagram also over here which says the same thing which we have already discussed. So I'll just be showing this to you. So over here you can see that C4A, it went away, C4B it stood, and C2A also it stood there, and C2B went away. So over here you can see that C3 convertase comprises of C4B, 2A. This is all about classical pathway. Over here you can see that C4 and C2, they get converted into C4A, C4B, C2A, C2B, and I've already told you that which ones move away and which ones stay there. So formation of C3 convertase is the place where classical pathway ends. Okay. Now let us move towards the next pathway, that is called as the lectin pathway. Let us talk about the lectin pathway. Now what is this lectin pathway? So lectin, actually on the surface of bacteria, some glycoproteins and polysaccharides are present. What does this mean? Suppose this is the surface of the bacteria. Okay, this is a bacteria and this is the surface of a bacteria. Now on this bacteria, these small proteins might be present, these small glycoproteins might be present, or polysaccharides might be present. So what are these glycoproteins or polysaccharides? They would be having carbohydrates. Now what are carbohydrates? It can be glucose, it can be galactose, it can be anything, it can be mannose also. Okay, so understand this, that whenever on the surface of the bacteria polysaccharides or glycoproteins are present and the last residue of this glycoprotein or polysaccharide is mannose. Suppose this is over here the last residue present, this is the last residue that is present, and suppose this residue over here is mannose, this residue over here is mannose, then what will happen? A particular protein is present inside your body that is called as lectin. What does lectin do? It identifies the end mannose present on these polysaccharides or glycoproteins that are present on the bacterial surface and binds with them or interacts with it. Okay, so on the first point over here it is written that it recognizes and binds to specific carbohydrate targets. Why are we saying specific carbohydrate targets? Because it is interacting with only mannose, and that too when mannose is present at the terminal, at the end. Okay, so carbohydrate target means mannose. What is binding to mannose? Mannose binding lectin. Okay, so why it is called as mannose binding lectin? Because it is lectin that is interacting with the mannose, right? So the second point I have already told you, bacterial surface glycoproteins and polysaccharides are present. Third point is it does not depend upon antibody for activation. Now if you have listened to me carefully, you would understand that I have never spoken, when I started lectin pathway, I never said that antibodies are required, or I never told that antigen antibody complex is required. Therefore it clearly says that over here antibodies are not required for the activation of this particular lectin pathway. So how does this get activated? Simply when the mannose is present on the end surface or on the terminal of the bacterial glycoproteins or polysaccharides, then this lectin that is present inside your body, it comes and binds, and this initiates or starts the lectin pathway. Now you might have a question in your mind that this mannose can also be present as a terminal sugar in our cases or in our cells. No, this does not happen. So this mannose as a terminal sugar is not present in mammalian cells, so it is not posing a problem. It will not create a problem in case of human cells. It will never be that human cell, because of a human cell the lectin pathway might get activated. No, it will not happen, because human cells, or for the matter of fact any mammalian cell, would not have mannose as a terminal sugar. Next point is part of innate immune system. Now since there is no antibody involved over here, that means no antigen antibody complex is not present over here. Antigen antibody complex is not present over here. Therefore this immune system or this lectin pathway, it is aiding the innate immune system. I have already told you that this innate immune system is actually a part of your is actually the immune system that you are getting from the beginning. Okay, it is by birth. Since no adaptive immunity part is present over here, no antibody is present over here, no T cell is present over here, this lectin pathway actually activates, or it is a part of the innate immune system. I believe this would be clear. Now let us move on to the next slide and understand that how does this lectin pathway function. Now over here you can see that this is a bacterial cell. Okay, this is a bacterial cell, and on the bacterial cells these are glycoproteins and everything that is present, and the terminal sugar that is present over here, this I am calling as mannose. Okay, now to this mannose what will bind? Yes, you are correct, to this mannose lectin will bind. That is why I am calling it as mannose binding lectin. Okay, now apart from the mannose binding lectin, two other proteins come to the aid. What are these proteins? These proteins are MASP1 and MASP2. Now MASP2 is actually just similar to C1s, the same C1s that you had just seen in the classical pathway. What did C1s do? Remember, it cleaved C4 and C2. Over here also this will do the same function because it is behaving like C1s only. So what will this MASP2 do? MASP2 will cleave C4 and C2. When it is cleaving this C4, it will form C4a and C4b. When it is cleaving C2, it will form C2a and C2b. I've already told you that C4b will stay and C2a will stay, whereas C4a will move away and C2b will also move away. I've written that also. C4a away, C4b stay, C2a stay, C2b away. So now what does this form? What do we end with over here? So since we are left with only C4b and C2a, so we can write it as C4b2a like this. And we have already understood this, we have already talked about this, that this is called as C3 convertase. Okay, why it is called as C3 convertase? Because it will act on the C3 and will break it into two parts, that is C3a and C3b. That is why it is called as C3 convertase. I believe you would have got this clearly, you would have understood this clearly. Okay, so if you could not see because of my this video, now you can see that I have written C4 getting converted into C4a and C4b, C2 getting converted into C2a and C2b. So this was written over here. Let me come back to my original position like this. Okay, so now let us move back to our discussion. Coming back to alternative or properdine pathway. This is the last pathway that we wish to talk about. Now before this, I would like to announce something that is very, very important. Now you must be knowing that GAT-B lectures or GAT-B batches are running from 10th of October. So if you are interested and if you are preparing for GAT-B examination, then you can get yourself enrolled in this batch that is running from the 10th of October. You don't need to worry about the classes that you have lost, because those classes would be available in the recorded. If I talk about the upcoming batches, this BPE live class or bioprocess engineering live class batch is going to start from November 5th. In this we would be talking about unit number 4 and unit number 5. The price of this course is rupees 1400 only, and if you add up the book that I've already launched a little while ago, the book originally costs around rupees 215, and if you buy it along with this BPE live classes, then there would be a discount and you would get both things for rupees 1600 only. Okay, so this is the announcement, and if you want to know more, or if you want to buy any of these things, you can directly get in touch with us on this number 8299134528. This is my contact number. You can directly get in touch with us, and you can also get in touch with us through mail or telegram. The details of this are mentioned in the description box. So this is the announcement that I wanted to make. Let me just come back to the discussion, that is alternative or properdine pathway. Now what is this alternative or properdine pathway? Let us talk about this. So alternative pathway, what does alternative means? Alternative means another way around or a different way. So this alternative pathway name came into the being after classical pathway was discovered. So it was taken as an alternate to the classical pathway. Why was it taken as an alternate to the classical pathway? Because remember, in the classical pathway there was antigen and antibody that were interacting with each other. Remember, there was antigen and antibody that were interacting with each other that activated the classical pathway. But when the scientists thought about this, they realized that there should be some other pathway also that would be activating the complement system without the aid of antibodies. So is there any system that can activate the complement system without getting help from antibodies? Yes. Then the name, or then the concept of this alternative or properdine pathway came into being. So the first point that you can see on your screen is it is antibody independent. Why? Because over here there is no antibody required. Next is, it is triggered by almost any foreign substance. Okay, so there is no particular, I would be talking about certain particular things through which it gets activated, but it can get activated by almost anything. For example, lipopolysaccharides from the outer membrane of the gram-negative bacteria. You would be knowing that there are bacteria that can be classified onto two parts depending upon the gram staining. So there is gram-positive bacteria and gram-negative bacteria. This gram-negative bacteria actually has lipopolysaccharides in the outer membrane. So this lipopolysaccharide can also activate the alternative pathway of complement system. Apart from this, certain yeasts, their cell walls can activate this system. Apart from this, cobra venom protein, apart from this, aggregated immunoglobulins and necrotic cells also can activate your alternative pathway. Now, since any kind of antibody or any kind of T cell is not involved in this pathway, so can you tell me which system, which portion of which part of the immune system does this alternative pathway activate? Yes, you are correct. It activates the innate immune defenses. Coming to the last point, over here actually 4 serum proteins are involved. What are the names of the serum protein? C3, factor B, factor D and properdine. This properdine is also called as factor P. You can see in the last line. So this properdine is also called as factor P. So what did we say? There are 4 serum proteins that are involved over here. C3. Now I have already told you about C1, C2. Over here C3 is also one of the complement proteins. Factor B, factor D and properdine. Now in the first slide only I told you that there are different naming systems. C1, C2, C3 is one naming system and the other naming system can be factor B, factor D. This is about the alphabets. Okay. So now coming to the next slide in which we would be talking how does a complement system get activated. Now over here you can see that C3 is present. Remember that C3 can automatically get broken in a very small quantity into its two parts, that is C3A and C3B. That means C3 is getting broken into two parts, C3A and C3B. Right. So what happens is that this C3B, what does it do? It can bind to some of the surface antigens that are present on the pathogens. Okay, so suppose the blue thing that I have underlined or highlighted is a pathogen, and on the pathogen there is a surface antigen. Okay, there is a surface antigen. So this is the surface antigen that is present. To this surface antigen, this C3b will go and bind like this. Okay, now this C3b is binding to the surface antigen. Now another protein, factor B, I have already told you that factor B would be involved. This is a serum protein. It comes and binds to this C3b. Factor B comes and binds to this C3b, and just following it, factor D also comes and acts on factor B. What did I say? Factor D comes and acts on factor B. So factor D comes and acts on factor B. So when it acts on factor B, it again breaks factor B into two parts, BA and BB. As I've already said that in general BB or the B portion, smaller B portion will stay and A portion will leave. So what we are left with, C3B was present in the first place, and after that BB has also come and joined in, C3B, BB. This is what we are left with. Now after the formation of C3B, BB, what happens is properdine comes and stabilizes this C3B, BB complex. Okay, so this C3B, BB complex is stabilized by properdine. Now let us come on to the activation of C3 and C5. So understand this, in all the cases what we have, in all the pathways what we have learned. So just before this we have ended the alternative pathway. So this is the alternative pathway, this is where the alternative pathway ends, that this C3B, BB is stabilized by properdine. This is where the your alternative pathway ends. Let us move on to the activation of C3 and C5. Okay, so if you remember, what did we study in classical and lectin pathway? We ended the classical and lectin pathway by the formation of C4. Yeah, so we ended the lectin and classical pathway by the formation of C4B and 2A, and we said that this C4B and 2A, it serves as C3 convertase, and when it acts on C3, it will break the C3 into two parts, C3A and C3B. Remember? So C3 convertase, what does it do? It cleaves C3 into two parts, C3A and C3B. C3B, it will bind to all the pathways. That means if I consider the classical and lectin pathway, C3B will come and bind over here. It will bind to C4B 2A, and the complete thing will be called as C4B 2A 3B. And if I talk about the alternative pathway, you would be knowing that we ended over here, C3B BB. This is where we ended, and now what will get added? C3B will get added because C3B BB was your C3 convertase. So the yellow, the one that I have already highlighted in yellow, this was C3 convertase, this was C3 convertase. This acts on C3 and forms C3A and C3B, whereas C3B over here will come and join with the previous complex. Now over here I will also be talking about what does the C3A do. C3A is actually a anaphylatoxin. What does anaphylatoxin means? It is basically mediating the allergic reactions, or you can also say that it is mediating the inflammatory reactions. What happens is C3A goes and interacts with the mast cells or basophils. Now you would be knowing that mast cells or basophils, they are having histamines. When these mast cells or basophils, they get degranulated, this histamine, it comes out. When this histamine comes out, it interacts with the smooth muscles. Okay, and in this process the vascular permeability, it increases and smooth muscle contracts. The smooth muscle will start contracting and vascular permeability will start increasing. So these are the features of basically allergic reactions. So it will be difficult, start getting difficult to breathe, and multiple other symptoms would start occurring, right? So this is how the C3 gets activated, or C3B binds to the previously present complex. Now understand this, after binding of C3B, what will happen? This will be called as this complex, this complete complex over here or over here, it will be called as C5 convertase. Why will it be called as C5 convertase? Because now it will be acting on C5 to again break it into C5A and C5B. C5A, again it will be released into the fluid phase because it will be functioning as anaphylotoxin, and C5b will bind to the cell surface. Which cell am I talking about? I am talking about the pathogenic cell. It will go and bind onto the pathogenic cell. So the C5b would be the first one to go and bind onto the pathogenic cell. Okay, now it forms the center for the binding of the terminal complement components. What does that mean? It will help in the binding of the next or upcoming other complement proteins. C5b will first serve as a docking site, and other proteins will come and bind with it onto the surface of the pathogen or cell surface that is to be destroyed. Now let us come to the terminal pathway. What do we understand by terminal pathway? So terminal pathway, by terminal pathway I mean that C5b is present initially over here. That means it has bounded to the surface of the membrane of the cell that has to be destroyed. Okay, so what are the components that are present in the terminal pathway? C5b is present, and just after C5b it will be followed by C6, C7, C8 and C9. This is just in continuation. C5b is followed by C6, it is followed by C7, it is followed by C8 and it is followed by C9. They interact or bind with each other sequentially to form macromolecular structure. That means all of these will bind with each other and they would form a macromolecular structure, a big structure that is together called as membrane attack complex. And this membrane attack complex would result in cell lysis. Now let us understand that how does this result. So just initially C5b was present, following C5b C6 came in, after C6 C7 came in, C8 came in and C9 came in. Now what is the activity of C9? C9 is actually a perforin like molecule. What does perforin do? Perforin creates pores on the surface of the cell. So if it creates pores, what will happen? Leakage will start and the lysis will happen. C9 does not exist as a monomer. It will bring about more and more C9 together at that place, and this would result in the polymerization. C9 will cause polymerization. This is about this. Now let me just finally talk about this. So suppose this was C5. Now you can see this C5b is present on the surface. C6 comes and binds to C5. Now C7 comes over here. Now you can see gradually they are penetrating the surface of the cell. After C8, C9 comes, and C9 comes in the form of polymers. Finally it creates a channel, it creates a you can see a pore like thing over here. With this, H2O or water starts coming in and ion starts getting out. So this causes actually leakage. This is the terminal pathway. Okay, now let us come to the functionality of poly C9. This is important, it can come in the examination. As I've said that this poly C9 will cause the formation of channels, transmembrane channels, and it disturbs the cell's osmotic equilibrium. That means it will change the osmolarity of the cell, because it is creating a transmembrane channel, and because of that multiple things will go out and come in. It will cause a leakage of the cell. Now ions will pass through the channel and water starts entering into the cell. Because of this the cell swells, membrane becomes permeable, and the ions etc. will escape from the cell, and this will lead to cell lysis. So this was all about the terminal pathway, right? So if we just brush up whatever we have studied, in case of the classical pathway and the lectin pathway, we talked about C4b2A, formation of C4b2A, and finally 3b got associated with it, C4b2A 3b. This was called as C5 convertase. C5 convertase added C5b to it, and C5b, after C5b, C6, C7, C8 and C9 came, and it caused the cell lysis. In case of alternative pathway. It started with C3b, BB, and after that 3b again came in, and then this will be called as the C5 convertase, then C5b will come in, and then again C6, C7, C8, C9. This will be the case. Now finally I would like to tell about the different functions that are mediated by the complement products. What complement or which complement proteins have what functions, this is what we would be talking about in this particular slide. So cell lysis, it is mediated by C5b to C9, that means C5b, C6, C7, C8, C9, they all are associated together to form the membrane attack complex. That is why we are saying that it is involved in cell lysis. Let us talk about inflammatory responses. So most of the A versions are involved in the inflammatory responses. So C5a, C4a, C3a, they are involved in the anaphylotoxin formation, or they are themselves anaphylotoxins. Chemotaxis of leukocytes, that means chemotaxis means chemically signaling the leukocytes to come at a particular place. This would be done by C3a and C5a. Opsonization is aided by C3b and C4b. Viral neutralization means neutralizing the virus, C3b and C5b to C9, the membrane attack complex. Finally, solubilization and immune clearance would be mediated by C3b. So this was all about the complement activation. This was all about the complement functionality, complement activation, and different functions that are mediated by the complement products. I hope this lecture was useful for you. You liked the lectures because we have covered everything in this just one video. I hope you liked it. I would request you to share this video amongst your friends so that all of them can get benefited. If you like this video, press the like button. Share my channel, subscribe to my channel, share it amongst your friends, and do not forget to press the bell icon. And yes, if you wish to join in any of our batches, I have already given you my contact details. You can get in touch with us on 8299134528. So this was all from us for now. Thank you so much and have a great day.